Hi everyone, my name is Lulu. I'm a graduate student in University of Michigan. Today I will be presenting our project PyLiger, Scalable Single-Cell Data Integration in Python. First, let's start with some background. High-throughput sequencing technologies now enable the measurement of gene expression, DNA methylation, and chromatin accessibility at the single-cell level. Integration of multiple single-cell datasets is crucial to the analysis of cell identity. Previously, we developed PyLiger, an R package that employs integrative non-negative matrix factorization to identify shared and dataset-specific metagenes of cellular variation. Here we present PyLiger, a Python implementation of Liger which enhances the processing efficiency of Liger. PyLiger also employs INMF to identify shared factors W and dataset-specific vectors Vi of cellular variation. These factors then provide a principled and quantitative definition of cellular identity. We redesigned the structure of PyLiger class to smoothly interface with the widely used NData format. Each single-cell dataset is considered as one NData object in PyLiger class. Cell factor loadings, share metagenes, and dataset-specific metagenes are stored as annotations in NData objects. The use of NData format also facilitates interoperability with existing single-cell analysis tools such as GLBIO and Scalable. In addition, PyLiger uses HDF file format for on-demand loadings of datasets stored on disk, providing us significant memory savings. Here, we demonstrate our PyLiger functions are 1.3625 times faster than their R counterparts on a PBMC dataset consisting of 6,000 cells. In particular, the most time-consuming step, matrix factorization, is approximately 3 times faster in Python than our previous R implementation. We also confirm that the factorization from PyLiger are identical to those from the Liger R package. Here is a visual confirmation. The UMAP coordinates differ slightly between Python and R UMAP versions. In summary, PyLiger is a Python implementation of Liger offering faster performance, optional on-disk format, and convenient interoperability with other single-cell analysis tools. Thank you for listening.